I will now introduce our next speaker. He's Dr. Abdelhadi Reba, aka Hadi. He's the group lead for the Replenisense program. Recently, he oversaw the discovery and preclinical testing of small molecule senolytics and biomarkers of aging. He will be presenting about a combination of senolytics and stem cell transplantation as a potential anti-aging therapy. You can read his presentation summary and bio on the event's website. Let's welcome him on stage and enjoy the presentation.
Hi everyone, thank you. I'm sorry for this delay. Thank you, Maria, for the introduction, and I would like to thank the organizers, including Maria, of course, for the hard work they put into this nice conference. I would like also to thank you all for being here. The project I will be presenting today is an ongoing project. It's one of the most recent project of SRF, and I will be giving you an overview of how we're approaching it and the progress made so far, and also I will be at the end talking about some next steps that we will do. Um, so I will just give you a quick background because most of you, if you have seen a meet and test talk yesterday, they give good details about senescence in aging. So here I just remind that yeah, it is one of the hallmark of aging, the accumulation of senescent cells, and they are implicated in many diseases and because they secrete pro-fibrotic factors, pro-inflammatory factors, and others that can influence the homeostasis of tissues. And there is a number of studies now that shows that definitely they are implicated in many aging-associated diseases. So based on this, it makes a good sense to target these cells for anti-aging interventions. In order to give you an overview of possibilities to target these cells, I thought that I can just put it this way by explaining basically the development from stem cells to senescent cells and in between. So most of the tissues have stem cells in them, and these stem cells, they give rise to progenitors and tissue-specific cells, and with differentiation or with damage or both, they end up becoming senescent, and even they undergo a cell death. The problem is that senescent cells are resistant normally to stress, so they tend to stay for a long time, and they don't really die immediately. And so they influence the surrounding cells, whether in the same tissue or in other tissues, by by secreting cells, and they can actually affect the organ function and they cause diseases. And one way to reduce the harmful effect of senescent cells is the most obvious one is to target them with senolytics. That can be either small molecules or immunotherapy. Um, Another way is to use xenomorphics. Those are molecules or small molecules or any other agent that can either inhibit the expression of SASP or reduce the harmful effect of SASP. Another way to target senescence is basically to inhibit or reduce the onset of senescence in the first place, and that's by targeting damaged pathways that cause senescence. This can be oxidation, can be other damages actually that can happen DNA or protein. One approach is to use any therapies that can or intervention that can reduce the damage. The only problem is there's too many of them, so but still it can be a possibility. Another one is a partial reprogramming. This is a new approach that have attracted a lot of interest recently, and it holds, in my view at least, it holds a good promise. The only thing is it needs to be optimized, and because you don't want to go all the way to reprogram cells to stem cells because of the risk of cancer. ideally is to rejuvenate senescent cells by reprogramming them. But that actually it's a little bit more challenging because senescent cells themselves are a barrier to reprogramming, but it can be possible. So yeah, this is a good approach to follow in the future. And of course, the one I will be talking about today is stem cell transplantation, stem cell, mesenchymal stem cell in particular. These cells have been shown many years ago that they can have beneficial effect when they're transplanted. They can have beneficial effect on many diseases, and mostly chronic diseases, bone, cardiovascular, degenerative diseases, and they were believed to act through basically just differentiating directly into tissue cells and replacing them, or at least replenishing them. But this hypothesis was not really supported by solid data. So the common belief right now is that mesenchymal stem cells exert their regenerative effect through secretion of factors, maybe mainly paracrine manner. So the secrete factors that can either, for instance, stimulate the proliferation and differentiation of progenitor cells. They can secrete factors that affect angiogenesis, immunomodulation, uh, and uh, and uh, and perhaps they may also act to inhibit SASP, almost like as xenomorphics. But that this is a possibility that we don't know a lot about it, and we are interested in exploring. So because mesenchymal stem cells have this good effects on various aging-associated diseases, and also they have a particularity of being safe. They have a good safety profile. So because of these reasons, we thought that if we combine them with senolytic, we may have a better chance in reducing the harmful effect of senescence. So with this in hand, the model we will be using is basically the naturally aged mice, because using genetically modified mice, for us, if we want to translate this to the patient, it's better to use a naturally aged mice. So in terms of treatment sequence, we are interested in we will actually do a senolytic first and then follow it with mesenchymal stem cells. And the reason for that is that we don't want to inject mesenchymal stem cell first and then senescent cells that exist in the body already will interfere with the function. And on the other hand, we don't want to inject mesenchymal stem first and then come with the senolytic, because that can disrupt the regenerative function. So the sequence is senolytic first and then mesenchymal stem cells. So the project actually will be in two parts. One is the selection, validation, and optimization of the senolytic themselves, which senolytic we want to use, and we will check we validate it and safety and everything. Also, we want to know which mesenchymal stem cell we want to use, the source, how they are optimized, and how we can make them survive. All these things we have to work them before even we try in vivo. And the most important things of all is the molecular markers of the treatment efficacy. How can we know whether senolytics or stem cells work without any markers? So we need to identify which marker to look for when we're using this intervention. And this is one of the big hurdles actually in the aging field, is to find the right molecular markers to study or to monitor interventions. After that, we will go to the next phase, which is uh, the actual testing of this intervention in mice. So we will do either senolytic alone, stem cell alone, or the combination. But for this other phase, we will do not only these molecular markers that we identified in the first phase, but we will also do physical and behavioral testing to follow up the health of these mice, and also we will record survival and health span of this mice. So this will be the phase two will be a little bit longer phase, but it's that's the goal of this project. So I will be in next few slides talking mostly about the optimization of these markers that we want to use. We want to know which marker to look for and where to look for them. So we are testing a variety of senescence marker. Isabeta gal, as you know, this is most standard assay for senescent cells. We will also be testing, we already start this assays, galactose linked to fluorescence dye, um, CY7 or galactose linked to Nile blue, uh, iron. We will also do immunohistochemistry with specific markers of senescence, P21, gamma H2X, P53, and we will also test ferritin because of the increase in iron senescent cells, EDO, marker of proliferation, and also some surface peptide that we have identified in the lab. We will run also doing PCR, ELISA for a variety of CESP molecules. And also we will do imaging for the markers, mostly the fluorescent markers, COA7 and gallium B. And also we want to track either using luminescence, uh, senescent cells labeled with luminescence derived from P16 mice. We also track mesenchymal stem cells labeled with several dyes. And we may also use the mice themselves, the P16 mice, to detect the burden of senescence. With regard to mesenchymal stem cells, we we will be testing them also the different markers. We're interested in survival markers because we want to know how long a mesenchymal stem cell when transplanted in mice, how long they survive in the body of these animals. We want to track them also by imaging, by labeling them and imaging them. The other thing we want to look for is also the function. Not only they need to survive, but also they need to be functional. Secreting factors that have rejuvenative function, the JAK, HIF, and others. And also we want to know whether when we are transplanted, whether the markers of the tissue that is affected by these cells, as for instance, fibrotic markers, whether they are reduced or not. So all these markers we are trying to optimize them in vitro, and we will also test them in specimen from mice, young and old mice, to see if they are working. So as an example of these markers, these are the standard one I just mentioned, the Isabeta gal. This is how it looks like in senescent cells. The EDU, it is expressed proliferation cells, not in senescent cells. Gamma H2X is labeled DNA damage. It shows basically a punctate in senescent cells, which detect the site of damage on DNA, and the P21 expression, which basically detect a cell cycle inhibitor. So it is increased in senescent cells versus non-senescent cells. So this markers seems to work very well. And we have also tested the CY7 gal, which is galactose linked to fluorescence molecule. When it is cleaved in senescent cells, it becomes fluorescent. This is a molecule that or probe that has been given to us, provided to us by a collaborator from Spain. It looks like it's working. It's selectively labeled senescent cells, but the signal is very weak. So that may be a problem when we put them in vivo, as I will mention later on. That can be a challenge. We have also tested the other marker, which is galactose linked to Nile blue. This one is more strong. It labels senescent cells more than non-senescent cells. However, it shows no specificity, probably due to a leakage. To maybe it's not very stable. So this also can be something that an issue when we move this type of markers in vivo. So we also translate some of these markers to tissue from compared tissue sections from young and old mice. And beta gal looks is working well. It's like a defined area in the lung that is expected to contain senescent cells. With regard to CY girl, again, as I mentioned to you, it's very weak signal. So but at least it does stain a little bit more in old tissue than young, but it's very weird. We also run a series of SASP expression molecules, and we can you can see that some of them are very good and have significant difference. In the red bars are the old mice, and the gray bars are the young mice. In particular, P16, which is known already as a marker for senescence, it shows very well here and it's working very well. It does consistently show a higher signal in old mice versus young mice. The other one, surprisingly, is serpin. It also has a significant decrease, although it has been restriction for in vitro to be increased the senescent cells. Here it shows a decrease, but it is significant, and that's fine as long as it is significant and reproducible. That those can be used as markers when we move to assessing the efficacy of interventions. With regard to MMP3 or MMP9, I think we may have to run increase the sample of the mice tested, and hopefully we will see more reliability of the staining. So this was in the liver, and if you move this, if you do the same thing in the kidney, the profile, the profile is different. And here we see that IL-6 was actually increased significantly in all the mice. MMP3, same thing, but P16 is not, and MMP12 included. So what this tells us is that when we will be looking for certain markers, we need to know which tissue to look for, or vice versa. When we want to look for marker in the tissue, we need to have this type of profile at hand so we can know which marker to look for in specific tissues. And also it makes sense because tissues have different cell type, and SASP should not be expected to be the same in every tissue. So yeah, this is an interesting finding that we want to develop further. We have also purchased this nice machine for imaging to image either the probe that I told you before, or we have some other idea in mind how to track senescent cells or stem cells labeled with different types that we are testing right now, and I will be able to share with you the result very soon. This machine also have been tested for the luminescence and for luminescence in P16 3MR mice, and it's working. So that's a good thing. So this is the advantage of using this kind of machine is to be able to basically monitor the efficacy of intervention over time without having to sacrifice the mice, and this is a good advantage. Another way actually to go about this is to try to find senescence biomarkers in body fluid. So let's say urine or saliva or serum. So this will be ideal actually for also for non-invasive longitudinal monitoring of intervention efficacy. This can be done, for instance, by using SASP in serum and detecting it by ELISA. However, this require blood draw, and when you are working with mice, you have a limitation of the volume of the blood to take, and this also will if you do it for a long time and several times, it will affect it will have effect on the results because it creates stress. The other way to go about it is to test CY GAL in body fluids. Our collaborator have tested this probe, and they claim that they can detect it in the urine within 15 minutes, and selectively increased in old versus young mice. But our finding did not confirm these results. And actually, we found the opposite. So this left us with the idea to try to look for some other markers that it can be detected in the fluid. So I have conducted some research on this and found an assay that is for fluorescence assay that has been shown for a metabolite that is secreted under certain stress condition. So we sought to determine if this metabolite is selectively secreted in senescent cells, and if so, can it be used to be detected, for instance, in urine from all the CMIs. So these are very preliminary data. So please bear with me, and you need to take them with a grain of salt. But we took culture media from senescent and non-senescent cells, and we measured this metabolite, and it was strongly induced in the culture media of mRNA senescent cells. I want to go a little bit faster, and I asked my colleague Marcela to collect urine sample from a couple of old mice that we have and two others young. And I told her, please don't tell me which one is which, and I will do the assay, and then we can see how it looks like. And sure enough, I was able to tell her 100% which one is which. And although it's only two mice, they are blinded, of course. And that was interesting. Even with two mice, it looks like it's significant difference. The bars are very low. But yeah, again, this is very preliminary data, so I cannot really. To me, it's too good to be true. So we need to do way more work to really confirm this finding. But this can be a really good essay for us, and we can do a lot of interesting things with it. If this work really, I call it senescence-related metabolite, and the associated pathway, we can do a couple of things with it. One is it can be almost like a like a diagnostic and therapeutic tool. For diagnostic part, we can test it, of course, in urine, plasma, and saliva, and see whether it correlates with age or with tissue with treatment intervention. And the other thing is we can target actually the pathway that leads to the secretion of this or synthesis of this metabolite. So we can target and hopefully we can find a vulnerability of senescent cells in this pathway. And also the other thing we can do is that use this, we can test if this metabolite itself have any effect on the microenvironment of senescent cells or their interaction with other cell types. So this is something that we are interested in, and we'll do some more work with it, and hopefully it will be positive. Now, of course, the molecular marker work is still continuing. We'll start with it, and we will continue for a while. But in parallel, at the same time, we're also testing senolytics and mesenchymal stem cells. So I will talk a little bit more about senolytics, but the last few years have been shown a lot of increase in discovery of new senolytics for the reason that I mentioned at the beginning, and they are the one that they discovered so far are not really they still have some limitation, mostly because most of them actually they are not selective, and some of them are cell type specific. Navitoclax is one of the most established senolytic in this whole series, and it has been proven even in our hand that it in fact kills selectively senescent cells, and it does also in some other studies in vivo. However, it have a toxic effect, mostly by targeting neutrophils, because they express the same target BCLXL that is targeted by that exists in senescent cells. So that's the problem. So that means that the translation of this molecule to the clinic will be a challenge unless there is an alternative or some derivation of this molecule. And our collaborator, that's exactly what he did. He took navitoclax and made a prodrug out of it. So basically, what he did, he added galactose to navitoclax. So this conjugate is called navitoclax galactose or just navgal, and so this navgal is inactive, becomes inactive, unless if galactose is removed in senescent cells by the galactosidase. So that means even if it goes in non-senescent cells, let's say neutrophil, for instance, this even if they have the BCLXL target of navitoclax, they will not be killed by it because they don't have beta galactosidase. So it will be active only in cells that have beta galactosidase. So yeah, that makes it very specific. It increases specificity much, and so it can be very safe. Hopefully, at least much safer than navitoclax. So we tested in our hand, in our cell model, and it does indeed kill selective senescent cells. A little bit less than navitoclax, and that was expected because navitoclax have only to cross the membrane of senescent cells to become toxic. However, navitogal not only has to cross the membrane, but also has to be cleaved by galactosidase in these cells in order to become active. But still, it's good enough, the activity that we see, and we bet on that for the selectivity and safety of this molecule in vivo. So we will be moving with navitoclax in vivo, and however, we always like to have a plan B, just in case that navitoclax does not work. So we are developing also a new class, sorry, a new class of senolytics prodrugs that are targeting a different pathway than just alpha beta gal. Sorry. So this pathway, as Tess has mentioned also yesterday, is the iron pathway. This has been shown. The iron has been shown to be increased in senescent cells and in aging for a long time ago. And this is a paper here that has been published in 2017 that shows actually, yeah, iron it increased by almost 30 times in senescent cells versus non-senescent cells. And we have published a paper last year that shows that confirmed that, and shows by labeling cells with this probe called siRNOx that it is stains mostly senescent cells and does not stain non-senescent cells. So it confirmed the increase of iron in senescent cells. And so we thought to leverage this increase of iron and in order to target senescent cells selectively. And for this, we are developing this novel prodrug that is activated only by iron Fe2 plus in senescent cells, and this I think will be hopefully more selective than the navitoclax or derived drugs. So with regard to stem cells, these are a cell population that actually it exists in various. It can be isolated from different tissues, and or even from iPS cells. And there are a series of markers that can distinguish them, and they have been shown to be able to differentiate in different tissues, in different cell types, in adipocyte, chondrocyte, and neurons, and muscle cells, etc. And the uniqueness or the advantage actually of these mesenchymal stem cells is that when they are transplanted, they have a beneficial effect and improve the outcome of various chronic diseases, including autoimmune, bone and cartilage, cardiovascular, inflammation, liver, muscle and nerve diseases. So because of this and because of all these diseases, most of them actually, they are aging-associated diseases. So it made good sense to try to see whether mesenchymal stem cells can have effect on aging itself. However, not much has been done in this area, and the only thing I can, of course, there is if you use hematopoietic stem cells, that's a different branch. But if you use like mesenchymal stem cells, there's only a few things that have been done. And this paper here, for instance, it shows that that if you use mesenchymal stem cells, particularly if they are primed or incubated, pre-incubated in normoxia, in 20% oxygen, they have more effect in extending the median lifespan of the mice. This is a RCC1 mouse model with damage sensitive. So yeah, this cells increase in the red line. You can see it does increase significantly the medium lifespan of mice, um, suggesting that yeah, if we can try to engineer mesenchymal stem cells or to find a good way to prime them, we may be able to have an effect on the lifespan and healthspan of the treated mice. The other thing that this figure here tell us is that mesenchymal stem cells normally do not migrate. They do not spread in the body. They tend to stay in the same area where they were injected. So and they don't survive for a long time. As you can see, probably 7 days, maximum probably 14 days, and that's it. So those are the area. If we want to improve the efficacy of these cells, we need to work in these two areas. One is the survival. We want them to survive first, and they will continue to circulate this molecule and have a regenerative effect. And the other thing, if we can improve the migration, the homing of the skull, that will be a plus. So basically, the consensus is that mesenchymal stem cells distribute a variety of factors that influence some specific area of the tissue. They can induce fact, basically immune cells. They can secrete chemoattractants. They can actually induce the proliferation of progenitor cells into neuron or other tissues. They can also induce angiogenesis, and this way they increase the supply of nutrient to the tissue, to the damaged tissue. So those are very interesting features. The only thing, as I mentioned, is that we need to at least to keep mesenchymal stem cells, transplanted mesenchymal cells, in the body long enough to have these effects on the tissues. So for this, we need to understand, at least how they die. And this is a paper that shows that when the label mesenchymal stem cell and inject them into the mice, they tend to go, as the other studies showed also, they go probably 7 days, maximum 14 days, and then they just die. And the way they die is by mostly by anoikis. That means they need to bind to something to substrate them in order to survive. Otherwise, they will just die. So we are trying to investigate this pathway, this anoikis pathway. Hopefully, we can find a way to make them live longer before the transplantation. So we have right now we are culturing these cells, we're characterizing them, making stocks of them, and also we are investigating survival factors, which one are maintained under certain conditions, and also the the functional markers we are also investigating them right now. So this is ongoing at the same time right now. So we're almost now done with these three parts, as I mentioned, molecular markers, senolytic and mesenchymal stem cells, and we're almost ready to move to testing them directly in mice. We will do either senolytic alone, stem cell alone, or the combination, and we will, as I mentioned, we will be looking for additional markers for behavior and survival. So the design of the experiment is straightforward, and basically for senolytic alone, for instance, it has been published. So we will do the same thing, and basically the mice will be treated for 1 week, and then for 2 days off, and then we start another week daily treatment. And then we will sacrifice the mice and collect tissues and do all these markers that I mentioned before. The primary outcome of this is to have a significant reduction of senescent cells and also the markers, including cells. And this is for us is limiting step. We need to find senolytic to move to the next step. So if this one for some reason doesn't work, as I mentioned, we are developing in parallel some alternative senolytics, and there are some others that we discovered in SRF before even I come. So we maybe have the opportunity to try this other senolytic too. So and after that we will also test senescent cells. Again, we are optimizing them right now, and we will take the most optimized ones, and then we will do the same thing. We will inject them. This is a single injection with the IP, and then after a few weeks, 4 weeks, we will sacrifice the mice and harvest all these markers and the tissue and test the markers. Here the outcome is mostly we'll be looking for pro-inflammatory and pro-fibrotic factors, secretary-based immune cells, as well as the expression of fibrotic markers on target tissue. So we will be looking into this, and again, if we will have to take the most optimized stem cells, we may also try EVs and see which one gives us the best read.
Hadi, I don't want to interrupt you. We have 4 minutes for questions, but you can choose if you want to answer the questions or you want to finish your presentation.
Yeah.
People can also find you in the room and ask questions. Just let me know.
Okay, this is almost my last
Okay.
yeah. Okay, so for the combination, I will be, as I mentioned, you will do senolytic first and then mesenchymal stem cell first. Here we will add some other markers of behavior, and we will do survival curves. And the outcome that we're looking for here is reduction of senescent cells and organ fibrosis. And we will be working on we will see hopefully the effect on physical and behavioral activities. And our primary goal, of course, is to see whether this treatment enhances health or lifespan of the treated mice. So, in conclusion, I just want to say that what we expect from this study at least optimize senolytic and mesenchymal stem cells, and this can be we can write some manuscript just on that, and the identification and potential synergy between the two anti-aging interventions. And we are also expecting to understand the mechanism, how the intervention interact with each other. And this will give us insight on selecting future intervention and all their combination to study a health span using the same process. And I just want to thank SRF donors and project sponsors, the SRF board and the executive team for all the support, and the research colleagues and the SRF staff. Also, I want to thank my team members and summer students and our collaborators for providing the material using this study. And thank you all. Sorry for the delay and for the technical problem that we have in the beginning. Thank you.
That was wonderful. Thank you so much. Also so interesting, really exciting. I have 2 minutes for a question. Actually, we have a question here. What is the average molecular weight of the senescence biomarkers?
Which one?
Did you hear my question? Can you hear me?
Yeah.
The question is, what is the average? Go ahead.
The average, you asked me about the average molecular weight of the senescence biomarkers.
Okay, there's
someone, but I don't know which one, which biomarker.
Average molecular weight of the senescence biomarkers.
Yeah, I will need to know which one. Yeah, and even that, I think some we know, some others we don't know.
Okay, and if you can speak a little louder for us, that'd be great.
Okay.
Hadi, we have one more question. I meant to say, I think it's about your metabolite.
Oh, the metabolite. Yeah. That's right now I cannot really talk about it too much because it's still very preliminary finding. I will definitely, I promise, I will give you all the details later. But right now, I just don't want to disclose too much.
I think we have 1 minute, but I see people typing, so I recommend that you find Hadi at his research booth after. Let's see. Last question, see if we make it. Chat keeps moving, I can't read it. Will you also have a treatment group that will have a senolytic plus MSC treatment at regular intervals from, for example, middle age onwards, as it is likely that periodically administering these treatments will have a greater effect than a single treatment cycle?
Yeah, it's possible. Yeah, definitely. I think we will at least for now we will use 18 months mouse. But later on, if it works and we want to do start earlier, we may consider this option. And but in that case, yeah, as the questioner asked, we will do probably more than one injection. Yeah.
Periodically. Okay, great. Well, we have to take a break. Thank you again, Hadi. Thank you everyone in the audience for watching these wonderful presentations that we're sharing with you. We're gonna take a 30-minute break. You can go and visit the other room through the teleporter if you want, or you can visit the booths that we have the wonderful students presenting their amazing projects. So go visit them, talk to them, and enjoy your break. I'll see you in 30 minutes.